A nerve impulse is an electrochemical signal used by neurons for communication. It is essentially a wave of electrochemical changes that travels along the neuron, facilitated by the movement of ions across the cell membrane. Neurons generate and transmit impulses through a series of steps. First, the neuron maintains a resting membrane potential. Upon stimulation, an action potential is generated. This signal then propagates along the neuron. Finally, the impulse is transmitted to the next neuron at the synapse. Resting membrane potential refers to the charge difference between the inner and outer surfaces of a neuron at rest. An unstimulated neuron maintains a potential of approximately negative 65 millivolts, though this can vary between negative 40 millivolts and negative 90 millivolts. This concept was first studied in squids. In neurons, the outside is more positively charged compared to the inside due to the unequal distribution of ions across the neurolemma. Sodium concentration is 10 times higher outside, while potassium is 20 times higher inside. This ionic imbalance is actively maintained by the ATP-driven sodium-potassium pump, which plays a crucial role in generating the membrane potential. The sodium-potassium ATPase pump actively transports sodium and potassium across the neuron's membrane. For every 3 sodium ions pumped out, 2 potassium ions are brought in. Since more positive charges leave the cell, the inside of the membrane becomes negative, while the outside remains positive, establishing the resting membrane potential. The negativity inside the neuron's membrane at rest is due to 3 main factors. The sodium-potassium ATPase pump, which actively transports 3 sodium out and 2 potassium in. The leakage of K plus ions through K plus leak channels causes further loss of positive ions. The presence of negative organic ions such as proteins and organic acids concentrated inside the neuron. These factors together create a negative polarity inside the membrane, and as a result, no conduction occurs at the resting state. As a key characteristic, neurons typically maintain a resting membrane potential of negative 70 millivolts, which is essential for their function. Action potential is the temporary reversal of electric charges across the neurolemma when the neuron is stimulated by external factors like touch, pressure, shock, sound, or vibration. This change in electrical charge occurs once the threshold potential of negative 55 millivolts is reached. The sequence of events in action potential involves depolarization, where the membrane potential moves from negative 70 millivolts to positive 40 millivolts. Repolarization, where the potential returns from 0 millivolts back to negative 70 millivolts. The initiation of a nerve impulse begins when a stimulus acts on the neuron. This stimulus causes a wave of changes in the resting membrane potential, RMP, as ions move across the neuron's membrane, ultimately generating an action potential. A subthreshold stimulus does not reach threshold potential, means if the stimulus is too weak to reach the threshold potential, no action potential will be generated, and the nerve impulse will not be transmitted. During the action potential, the process begins with the opening of sodium channels, which causes an influx of sodium ions. This results in the membrane potential changing from negative to 0 millivolts and then progressing to plus 40 millivolts. At this point, the sodium channels automatically shut down and potassium channels open, allowing potassium ions to move out of the cell. The outward movement of positive charges restores the membrane potential back to a negative value around minus 65 millivolts. To maintain this ion distribution, the Na-K plus ATPase pump is activated, restoring sodium to the outside and potassium to the inside of the neuron. Once generated, the action potential spreads locally along the neuron, traveling down its entire length. This wave of electrical activity ensures the transmission of the nerve impulse. In the nervous system, communication between neurons happens at specialized junctions known as synapses. A synapse is the region where impulses move from one neuron to another. The synapse consists of 3 main parts: the presynaptic membrane, synaptic cleft, and postsynaptic membrane. The presynaptic membrane, found at the axon terminal, releases chemical messengers called neurotransmitters. These molecules travel across the synaptic cleft, a narrow gap that separates the 2 neurons. The neurotransmitters then bind to receptors on the postsynaptic membrane, triggering a response in the next neuron. Interestingly, if the postsynaptic neuron is a muscle, this connection is called the motor end plate, which plays a crucial role in muscle contraction and movement. Neurons communicate with each other through specialized chemical messengers called neurotransmitters. These molecules are stored in small vesicles within the axon terminal. When a nerve impulse reaches the axon terminal, calcium channels open, allowing calcium ions to flow in. This influx triggers the release of neurotransmitter-filled vesicles into the synaptic cleft. Once released, neurotransmitters travel across the cleft and bind to specific receptors on the postsynaptic membrane. This interaction generates an electrical signal in the next neuron, continuing the transmission of the impulse. There are different types of neurotransmitters. Excitatory neurotransmitters, like glutamate and acetylcholine, promote signal transmission, while inhibitory ones, like GABA and glycine, reduce neuronal activity. Others, like serotonin and dopamine, play key roles in mood and behavior regulation. This process is chemically significant because blocking transmission at this site with drugs can promote muscle relaxation during anesthesia. Additionally, it helps reduce muscle spasticity in certain neurological conditions. Reflex action is a sudden and involuntary response that occurs either due to an internal or external stimulus. It is an automatic reaction that does not require conscious thought. Common examples of reflex actions include withdrawing the hand from a hot object to prevent injury, the knee-jerk reflex observed when the patellar tendon is tapped, and the blinking of eyes to protect them from dust or bright light. A reflex arc is the neural pathway involved in a reflex action. It typically consists of sensory and motor neurons, transmitting signals rapidly to produce an immediate response. There are two types of reflex arcs. In a monosynaptic reflex, the pathway includes only sensory and motor neurons, as seen in the knee-jerk reflex. In a polysynaptic reflex, interneurons are present between the sensory and motor neurons, allowing for more complex processing. In vertebrates, most reflexes are polysynaptic. This means that, in addition to producing an automatic response, signals are also sent to the brain's cortex, enabling some level of voluntary control over the reflex. Saltatory conduction is a process that occurs in myelinated axons, where the nerve impulse jumps from one node of round VA to another instead of traveling continuously along the axon. This jumping mechanism significantly increases the speed of nerve impulse transmission and improves efficiency, allowing faster communication between neurons and target organs. That concludes our module on nerve impulses. In the next video, we'll explore the nervous system. See you there.